Bismillahirrahmanirrahim. How are you guys? Hope all you will be good. My name is Yaul. I'm from GM first 4th semester. So today my topic is in pharmacology about the antihyperlipidemic drugs, cholesterol absorption inhibitors, and the last one, bile acid sequestrants. So before we really start the topic, let me clear you something in advance, like what is LDL and what is HDL. So low density lipoprotein, as name indicated, is a combination of the lipids and proteins. So these they transport the cholesterol or lipids from liver to the tissues. So we know already that if in the tissue abundance of cholesterol occurs, so clot formation and the stroke will be occurs. So that's why they are called bad proteins. Next we have high density lipoprotein. So high density lipoprotein, they are called good proteins because they absorb back these cholesterol from the tissues and store it into the liver. So that's why they prevent us from the risk of the heart attack and the stroke. So that's why we are called good protein. Let's come towards our topic, which is called antihyperlipidemic drugs. Before we really start it, I want to tell you something like as an introductory, antihyperlipidemic drugs. It is also called cholesterol lowering drugs. It is also called cholesterol lowering drugs. Like it is a diverse group of pharmaceuticals that are used in the treatment of high level of fats or lipids such as cholesterol in the blood. So if excess amount of cholesterol occur in the blood, in this condition, we are called hyperlipidemia. So they are called lipid lowering drugs. So as name indicated, anti-hyper, so against the lipids, the drugs which we used, they are called antihyperlipidemic drugs. So these are the introduction of the antihyperlipidemic drugs. Next one, the most important thing before we really study the cholesterol absorption inhibitors, we have to learn a brief concept about the cholesterol. That what is cholesterol? So cholesterol is actually a waxy fat-like substance found in our cells, in our body. Cholesterol is a fatty substance found in our cell and our body. And if you talk about its functions is to make different types of hormones, like it also help in making the vitamin D. It also help in making some other substance which help in digestion of food. If you talk about its chemical formula, so generally it have 27 carbons, 46 hydrogen and a single oxygen. So next, cholesterol, as you see in the diagram, they help in regulating and synthesizing a lot of hormone like the cortisol, the progesterone, and other steroidal hormone like sex hormone. In sex hormone we have the testosterone, estrogen, progesterone. So they help to secrete that type of hormone. And the hormone structure, you see the formula, the cortisol and glucocorticoid, the structure of glucocorticoid and cortisol you see here. So cholesterol and phospholipid combined together, they form also help in making the cellular structure, the cell membrane. They help in supporting the cell membrane, these two together. If you talk about the phospholipids, so they have phospholipids have two types, like a head and a tail. As you know, the upper border portion and the round portion is called head, and the lower tail-like portion is called tail. So head are generally hydrophobic, means they hydrophobic are those which can't love water or accept water, and the tail are hydrophilic. Hydrophilic are those which accept water. So these are the general introduction about the cholesterol. Now we are in position to talk about the cholesterol absorption inhibitors. So which type of drugs we have to take and we have to use if we have abundance of cholesterol in our body? So we have to use the ezetimibe. It's one of the drugs which used as a cholesterol absorption inhibitors. So now we are in position to study about the mechanism of the action of the ezetimibe. So it is a class of compound that prevents uptake of cholesterol from a small intestine into the circulatory system. Here you can see the small intestine. So ezetimibe, it is a drug which inhibits the cholesterol transport protein from the, here you can see the NPC1 and L1, Niamin, PIC C1 and LICA1. These are the protein which act as a cholesterol transporter protein. These help to in the transportation of cholesterol, NPC1 and L1. These are the protein which act as a transportation of the protein. So they generally prevents absorption of free cholesterol in the small intestine. They generally inhibit the NPC1 and L1 protein and prevent absorption of free cholesterol in the small intestine. They also act another type of action, like they also inhibit or block the AP2 enzyme. They also block the AP2 protein. So AP2 protein is generally responsible for the endocytosis and endocytosis of cholesterol from the small intestine to the blood. We have two types of mechanism, like one is endocytosis and the other is exocytosis. So endocytosis are the eating of cell and pinocytosis are the drinking of cells. So they stop in this way. So in this way, ezetimibe block absorption of cholesterol from the small intestine to the blood. So next we have the ezetimibe. So interior dose of ezetimibe, and brand name is Zetia. So interior dose of ezetimibe is 10 milligram, and they didn't have any parental dose. Interior dose are those which follow the GIT, gastrointestinal tract. The drug which we use it orally, so we call it anterior doses, and half-life is about 22 hours. Ezetimibe drug have a half-life of 22 hours. Next, we have a second type of, they also block the PCSK9 proprotein convertase subtilisin, or we also call Kexin type 9 inhibitors. So they also block the these enzymes, and if these enzymes are blocked, so they come here and block the low density lipoprotein receptors. So here two types of substance you can see here. These are called antibodies, alirocumab and evolocumab. These two are antibodies. They also block the PCSK9 enzyme. So if this enzyme are blocked, so they prevent us from degradation of low density lipoprotein receptor. So then what occur? The greater amount of low density lipoprotein receptor and catch more LDL, because concentration here is low, so they catch more LDL from the circulation of the blood. They catch more LDL from the blood and decreases the low density cholesterol level in the tissue. So this are about the mechanism of the ezetimibe while blocking the PCSK9 enzyme. So here you can see the alirocumab and the evolocumab. It has and we didn't take it in interior doses because they have a parental dose. So parental dose are those which have to be taken while other than oral, like intramuscular, intravenous, intracutaneous, rectal route, buccal route, and so on and so forth. So they have a high bioavailability and they have a first pass metabolism. So the half-life of the alirocumab is 17 to 20 days, while they have a 75 to 150 milligram per milliliter. And the evolocumab have a parental dose of 140 to milligram per milliliter and to 420 milligram per 3.5 ml, and the half-life is about 11 to 17 days. So next we have the our last and the third topic, like bile acid sequestrants. So before we start it, we have to introduce a little bit the bile acid sequestrant. That what are bile acid sequestrants? And in this table you can see here the mechanism of bile acid sequestrants. So bile acid sequestrants are medicines that help us to lower your LDL, low density lipoprotein cholesterol. So too much cholesterol in your blood can stuck to the walls of your arteries and narrow or block them. So these medicine work by blocking bile acid in your stomach from being absorbed in your body. So if you have too much cholesterol, so if you take these type of medicine like the class of bile acid sequestrant medicine, so they help in prevent you from blocking the blockage of and get you free from the blockage of the cholesterol in your narrow arteries or veins. So these are the introduction. So bile acid sequestrants generally block the reabsorption of bile acid from the gut. Here you can see they block the reabsorption of bile acid from the blood, from the gut. So if we don't have bile, so liver will increase the secretion of bile acids, and this way they comes to the gut and absorb the substance if they block by some purpose. So bile acid generally made up of cholesterol. Its composition like they are made up of cholesterol. So low density lipoprotein receptor or cells generally increases the number of low density lipoprotein receptors to bring greater number of low density lipoprotein cholesterol. So as a result what will occur? As a result the less level of the circulatory LDL and LDL cholesterol in the blood will be occur, and we get us free from the cholesterol in our blood. So these inhibitors generally block not to store the cholesterol in the blood, so we can the RBC will move freely and we didn't pass any clot. So we also stop the bile acid naturally. Like what we have to do if we want to block the bile acid. So firstly we have to stop the smoking. Next we have to eat simple meal. Next limit fatty foods, like we don't have to eat the shawarma or burger etc. like that fatty substance food, and avoid alcohol or loss in the excess weight. We have to proper exercise. So these are the topics, and these are all the topics we study about the antihyperlipidemic drugs, the cholesterol absorption inhibitors, and bile sequestrants. So hope you get some idea from it. Thank you.